the brain, okay, as it relates to the brain, we'll come back to this again, but that's going to be related to what? Your mood, yeah, motivation, what? Attention. Ah, yeah. Attention, motivation, mood. So it makes you feel good. Makes you feel good. Makes you think well. Good memory. All those things. See? Could that be addictive? Yeah. Yeah. Okay, there we go. Yeah, so, but what else does he do outside the brain? Activity for all those other things. What? Vasoconstriction, smooth muscle, activity, yes. Heart, cardiac muscle, pump up the blood. Yeah. If I increase my heart rate and I increase my blood pressure, before when you came to the ER and you didn't have a pulse, you think you're going to have one now? Yeah, that's why we put that 1 milligram in there. Okay? And now, oh, you got a pulse now? There we go. Now I'm happy. Now let's work on this patient. See, you use epinephrine for a lot of other things too, not just the brain. Okay? But he's in those drugs that are related to the brain. Okay? He's in those drugs. But those are your what? Catecholamines. Catecholamines. One who's a biogenic amine, not a catecholamine, he's helpful. What's his name? Serotonin. You know. See, the situation with our different neurotransmitters, even the one we started with, when they bind to their receptors, they're not going to stay there. What happened to cyclic AMP? Remember that second messenger from chapter 5? What happened after he actually did what? After he activated a particular what? What does he activate? A protein kinase. A specific. When he activates him, does he continue to? He doesn't continue to? Why doesn't he continue to activate protein kinase? Is there something, when he goes across that Hudson and binds to his receptor and activates that protein kinase, does he stay there with that particular protein kinase? Who's there? There's a shark right there to do what? Grab them, break them up. And that's why you don't have a continuous response. See, that cyclic AMP continued that sequence of events, made sure a cell had a response. You don't have a continued response. Because you always have these ways in which that messenger, he's going to come out and get out of the picture. There's going to be that shark, boom, take him out of the picture. That's good. You don't want to have continuous stimulation, right? And so for acetylcholine, we have some shark that's, he has a shark, right? And his shark, okay, is called what? What's his shark called? His shark is called acetylcholine esterase. See, ASE, it's an enzyme. It's an enzyme where? Here. In that interstitial fluid, and as soon as he binds, what does that shark do? Grab him, chop him up. All right? So that way he causes our actual what? It causes either an excitatory postsynaptic potential or inhibitory postsynaptic potential. But not for what long? Okay. If it's a message that is going to be continuous, got to be another neurotransmitter coming behind it, okay? Because he's going to constantly be what? Taken out. There are actually 3 ways in which a particular neurotransmitter is actually going to go away. 3 ways, all right? One way, okay, in which he goes away is through diffusion. What is that? You had your neurotransmitter crossing the Hudson, but what happened? He drowned. Okay, that was he actually bound to that receptor, but his actual concentration went away from that receptor. Why? Diffusion is when you're at high concentrations somewhere, And then you want to go to the what? The low. And he just disappears into that fluid. Okay. So that's diffusion. That's one way you're not going to have constant stimulation. The other way is what we just mentioned, which is what? Your enzyme breaks it down. There's an enzyme, right? ACE. There's an enzyme in that actual Hudson River that actually takes your actual what? Your messenger out of the picture. Enzyme degradation. Okay? And then the last way is that you'll have certain neurotransmitters that actually do what? Go back into the actual what? Presynaptic neuron. Okay? They go back to where they came from. Is that okay? So we just call that what? Reuptake. Okay? We call that what? Reuptake. 3 ways in which you will not have constant what? Stimulation. 3 types of ways there will be a fate of that particular messenger. 3 actual ways in which your enzyme, okay, has their fate. Or your particular messenger has its fate. Is that right? 3 ways. Which way? What's the what? Diffuses away. Enzyme breaks it up. Or it goes right back into the cell. These guys here, they're going to go back into the cell. Okay? Your actual catecholamines, the way in which he's actually going to go away from that receptor is because what? Goes back up into the cell. So I can give a drug if I want to increase my dopamine. If I want to do what? Increase my norepinephrine or increase my epinephrine and keep it on that receptor. Why would I want to do that? Well, because it makes me what? Feel good. Because it makes that patient what? Feel good. Makes that patient think that way, think better. Is that right? Can you name a type of patient who you want them to actually have that better memory and better attention? ADHD. All right. So I have a drug that's going to stop that enzyme, right, for these guys. That enzyme for these guys is called monoamino oxidase. Monoamino oxidase. He's going to take out those guys. Okay. Monoamino what? Oxidase. So I'm going to put a drug in that inhibits the monoamino oxidase. Okay. I'm going to put in a drug that's a monoamino oxidase what? Inhibitor. Okay. A monoamino oxidase what? Inhibitor. Yeah. And so basically now you feel that way. No problem. Very good. Okay. You know, as far as serotonin, what are we going to use? Well, he's reuptake. He goes right back into the cell. But I can put a drug in that stops him from going through the process of being what? Reuptaken into the cell. Is that right? So we call those drugs exactly what do we call those drugs? That's exactly what we call them. We're stopping the process from them to what? Be retaken into the cell. We call them what? Serotonin reuptake inhibitors. Is that okay? But they're selective for who? Serotonin. So we say selective serotonin reuptake what? Inhibitors. SS what? RI. Is that it? Right? Can anybody name one? One's called fluoxetine, but you ever heard of Prozac? Okay. All right. Sertraline's another one. Okay. Sertraline. There's different ones. And what are they going to help with? Serotonin really makes the mood what? Feel good. Right? So what kind of patients I might give that to? Depression. Okay, so that's a big one. All right, those pharmaceutical companies are doing extremely well with that one. Okay? So yes, serotonin makes you feel good. That's in the brain. Outside the brain, actually it's mostly outside the brain. You're actually going to find that he does things like vasoconstriction, right? Activating those cells that do certain things. So increasing blood pressure, digestive system stuff, he inhibits things. Well, we'll come through that when we get to it. We'll see him a lot too in other systems. Right? So yeah, you have your different, your different neurotransmitters. Let me pay attention. When we look at acetylcholine, because he can do both, that exciting process and inhibit. But if we focus on the exciting, he can do a lot of good exciting things in the brain. So that way you can think well. All right? And that's the guy who's going to cause a problem when you get over 65 years old. What's our concern? What kind of disease that you're concerned about over 65 years old? Yeah. And let me tell you, 15% of the population over 65, they get Alzheimer's. That's 15%. What if you had a 15% chance in walking in this room to get in, you get shot? Would you want to get into that? That's a high, not 2%. 15%? Right? What's the chance? Over 85, would you read as far? If you're 85 years old, what's the chance that you'll have Alzheimer's? 50%. That's just flipping a coin. You might as well get 85 when you turn 85. Look forward to it. Look forward to what? Not knowing where you are. You just drove to the store and you just said, I don't even know where I am. How about that feeling? You want that feeling? Well, you can't remember your children. Don't know where you are. Don't know the children. And there's flashes of that. Right? That's got to be scary, right? Yeah, this is terrible stuff. So, they do have certain medication that will actually try to get rid of who? Try to get rid of this guy. Is that right? If you get rid of acetylcholinesterase, then you're going to increase the amount of acetylcholine doing what? Binding to his what? Receptor. Is that right? See, acetylcholinesterase takes them away. So if we actually give some drugs going against that enzyme, then boom. Okay. Remember that phosphodiesterase? Who got rid of the phosphodiesterase? It was yes. We'll say it loud. Remember, caffeine took out the sharp for cyclic AMP. You remember? And that's why cell kept going, right? That's why you feel that way with caffeine. Okay. So you have drugs that kind of help with Alzheimer's. Because the point is, what Alzheimer's? The acetylcholine is what? Love. Those neurons that produce acetylcholine, love, right? So, they sometimes it's not there yet. So a lot of research needs to be done in that, right? But my people going into medicine, that's a place to go to. We need help with that. Especially help me, right? Look, I'm closer to the 65 than all of you guys. Okay, come on. Let's get something for this. That's terrible, right? So yes, okay, we have our different neurotransmitters. And when we look at, and this is one neuroglial cell I want to end with, right? Because what I like to go back to is conduction, right? Conduction. How a particular action potential moves down the neuron. That is important because that's related to how fast you're going to have an effect. See, it's the neuron that conducts that message, that signal to the next place. Right? So that matters how fast it is. Right? How fast that is. That tells you how well conduction is. And so we have this supportive cell. What are the supportive cells called in the nervous system? Neuroglial cells. See,